Good afternoon, everyone. I would like to welcome you to today's talk, which is on cancer immunotherapy. Our speaker today is Sergio Quezada. I hope I pronounced this well with my Brazilian accent. And he joined UCL in 2011 as head of the Immune Regulation and Tumour Immunotherapy Group. More recently, Sergio has held the CRUK Career Development Fellowship. After his talk, we are going to be taking questions from the room and also from the online audience. The Twitter feed for the lunch hours is UCL LHL, and today's event code 2315. And now with Sergio Quezada. Thank you.
Thank you very much. It's a pleasure to be here, and it's actually a big task to try to explain to a general audience what's going on with the immune system and how we can benefit from mobilizing this complex army of cells against tumors without actually damaging the host. So I think the most important thing is to start with a little bit of background. And the background, the reason I usually show this slide is because perhaps some of you guys have heard how important is the immune system in the last 5-6 years, with some successes on clinical trials and lots of articles in the paper saying that immunotherapy will help us cure cancer, et cetera, et cetera. And the point of this slide is to show that it is true that there has been a lot of excitement in these last 5 years or 6 years, but they are not the result of something discovered in 2010. Absolutely not. So this is the result of more than 100 years of research, and there are some key case studies or examples of things or discoveries in terms of immunotherapy or cancer immunology that have led to this final set of data that suggests that the immune system is actually a powerful tool against cancer. So if we go to the very beginning, there's this gentleman, William Coley, which actually is kind of one of my biggest heroes in cancer and in medicine. I'm not a doctor, I'm a mouse doctor, but this doctor is a surgeon. So William Coley is a surgeon from Memorial Hospital in New York City, which today it's called Memorial Sloan Kettering. He was a bone sarcoma doctor and a very observative scientist also. So he made a very key observation a long time ago. He realizes that patients that develop sarcomas, which are very nasty tumors, that got infected, they spontaneously regress their tumors. So infection correlated with regression. He obviously did a large follow-up of many patients undergoing this. And then he had the brilliant idea in 1891 to take one patient and inject their tumors with heat shock bacteria. Because his hypothesis based on his observation was there was something unknown that was being activated by bacteria that allowed the elimination of tumors. And over the next 40 years, he treated more than 1,000 patients with heat inactivated toxins, which is called Coley's toxin. And he cured several of them. Now, interestingly enough, his colleagues never believed him, right? This was, again, in the late 1800s. Nobody believed him. And at the same time, radiation therapy, because of the work of Madame Curie, came up to age. So Memorial Sloan Kettering decided at that point, early 1900s now, to stop all the work that Coley was doing and focus on radiotherapy. So in a way, radiotherapy killed immunotherapy for about 50 years. Now, the key thing from this is that we now understand that what he was doing in his clinical trials, it was actually mobilizing an immune response. The immune system realized that there is an infection, and because it was an infection at the tumor site, it mobilized the whole immune compartment into that area, and that in some cases produced anti-tumor activity and tumor destruction. So William Coley is considered the father of cancer immunotherapy. So I think what I'm trying to say here is that the immune system can be mobilized to fight cancers, and that's key. Now the problem, if it can be mobilized to fight cancer, why is it failing from the first instance to protect us against cancer? Why is it failing? Why is it not protecting us? And that's where we come up to another key luminary in the field, which is Sir MacFarlane Burnet. And basically, Sir MacFarlane Burnet had the following question. Is our immune system really bad at killing cancer? Right? Is it really, really poor at killing cancer? And what MacFarlane Burnet hypothesized, he generated a theory called the immunosurveillance hypothesis. And I'm not going to read it because I don't have a proper English or British accent, but if you guys read it there, it will say that as a malignancy develops, it will have antigenic determinants, strange things, that might induce an immune response. And that results in a non-evident disease. In other words, if we're walking on the street cancer-free, that's because my immune system is attacking. So my immune system is constantly surveying. And that hypothesis was, again, argued for more than 30 years. And it has only been on this last decade that it has been accepted as true. Now, one of the issues is that if the immune system can provoke a response and eliminate tumor, if there is such a thing as immunosurveillance, then why do we have people with cancer, right? When is our immune system failing? I'm trying to tell you that it's very good, but it's obviously failing because people that we know develop cancer. Why does it fail? Well, the problem, which is actually a good problem to have, is that the immune system lives on a tight balance, right? So you can imagine this being your immune system and this being yourself. If you have the perfect balance, you are protected from nasty bacteria and all that evilness that exists there. If your immune system is depressed, is not functioning, then you fall in the pool size, right? So it's a bad thing. But also, if you have a very active immune system, that doesn't look pretty at all, right? It looks very ugly. So you want to live at this level. You want to be happy and well-balanced. You don't want an underactivated or overactivated immune system. So tumors take advantage of this regulatory system. They take absolute advantage of it, and that's why, in a way, tumors develop, or one of the many reasons why tumors develop. So we'll try to go into this to try to understand regulation, how does it work and how the tumor takes advantage of it, but for that we need to go back into the basics. So this is an overview of the immune system. So what everybody says, it's an army of cells, blah, blah, blah. There will be a quiz at the end of the talk, so just memorize all of them. So I am a T-cell chauvinist, so I'm just going to show you the cells that I think that for today's talk matter, right? Not the cells that the only ones that are important, but the ones for today. So which ones are those? The tumor, right? The evil. The APC. The APC is a specialized immune cell. APC stands for antigen-presenting cell. I'll explain you what it does, but that's the APC or dendritic cell. And then our good guys, the T cells. Now, why am I so focused on T cells? Why do we think that T cells are so relevant? Well, there are lots of studies like this one. This is a survival curve in patients with metastatic melanoma. And what it shows is that patients that have a high number of T cells infiltrating their tumors, they survive longer than patients that have a low number of T cells infiltrating the tumors. So T cells stand for terrific. They're very good to have inside your tumors, okay? Now, back to the regulation. If T cells make it to the tumor, why we still get cancer? Right? So they get there, but there is something failing. And then that's where the other two, actually the modern fathers, or the fathers of modern cancer immunotherapy come to play. So Bob Schreiber and Lloyd Old, they follow up on the work from McFarland Burnett, and this is on the early 2000s. And what they managed to do using proper scientific method and lots of animal models, they managed to demonstrate that actually immunosurveillance does exist. And they proposed the following hypothesis now based on a lot of data. It's called the 3 E's of cancer immunoediting. What they say is that as a normal cell accumulates mutations, right, it will get those mutations that will transform that cell. And then that cell will become a cancer. At the first instance, and this is the immunosurveillance hypothesis of McFarland Burnett, that tumor, that developing tumor, will have antigenic determinants, strange proteins on their surface, proteins that will look foreign to the immune system, and the immune system will go and eliminate that tumor cell. So that's immunosurveillance. That's the first E of immunoediting. That's why we walk around on the street without cancer, right? The problem is that if the immunosurveillance fails, you develop a tumor. And that tumor enters on a phase of equilibrium. And this is a relationship, like a very damaging relationship between the tumor and the immune system. The immune system tries to attack the tumor fights back, the immune system attacks, and it fights back, and generates that balance, right? That could be a nevi or a pre-malignant lesion. And the problem is that when the tumor wins that battle, using different mechanisms of immune evasion, you have the third E, which is escape. And that's a full-blown growing tumor that ends up killing the patient. Now, where does regulation start? It starts at this level. So as a tumor turns over, dies and grows and dies and grows, bits and pieces, antigenic determinants of that tumor come out and they float in the periphery. And then this specialized cell, the APC, is able to pick up those pieces of tumor. And it displays them on a machinery that is called the antigen-presenting machinery. And it literally presents those pieces of tumor to our terrific friends, the T cells. And actually, if we want to complicate that a little bit more, and we want to look at that interaction between the tumor and the T cell, sorry, between the antigen-presenting cell and the T cell, it looks like this. And this is key. This is the antigen-presenting cell. This is the T cell. The specificity of that interaction is given by a molecule called the T cell receptor, or TCR. So when the T cell receptor recognizes something strange on the antigen-presenting cell, something that comes from a tumor, the T cell stops. Well, let's say the T cell stops and looks up and says, okay, what is this thing? This thing is strange. It doesn't look self. So it must be a tumor, or it must be a virally infected cell. But that's not sufficient. That's called signal 1. That doesn't activate fully the T cell. And what the T cell requires is inflammation and danger signals. And when you have those inflammatory and danger signals, is that you get this, more specifically, co-stimulatory signal called signal 2 through a specific receptor. And that's when the T cell gets fully activated and fires away trying to find the tumor and destroy the tumor. So signal 1 makes you stop and think. Signal 1 plus 2, it makes you act against the tumor or the virally infected cell. Unfortunately, nothing in immunology is ever that simple, so I'm going to complicate the story even more. So a point of inflection on this more than 100-year trajectory is the discovery of a very important molecule called CTLA-4. Right? This is a molecule that was discovered to be expressed on the surface of T cells, of lymphocytes. For about 7-8 years, that's all we knew, that it was expressed on T cells. The function of that molecule was not known. And then it was in 1996 that this fella, Jim Allison, he proposed and actually demonstrated on a science paper that this molecule acts as an immune checkpoint. What does it mean by immune checkpoint? He says that this molecule is expressed on T cells that are trying to build up an immune response, and he shuts them down, just to make sure that you're not going to go crazy trying to destroy any cell. And what happens is that the tumors take over that mechanism. And we'll go deeper into how does that work. And then it was about 20 years later or more that, and a lot of science, and a lot of basic science, and a lot of clinical trials that led to the approval of the same molecule he generated and he tested in mouse models of cancer to its approval against late-stage metastatic melanoma. So a long promenade, right, of work to take us from a molecule to a mouse model of cancer where he demonstrated that antibodies that blocked that molecule could eliminate the tumor, to the approval of a drug. This is how CTLA-4 works. So again, the antigen-presenting cell and the T cell, you get signal 1 through the T cell receptor and signal 2 through this co-stimulator. CTLA-4 accumulates within the cell in intracellular vesicles. And at the moment that the T cell looks up and stops to think whether to act or not, CTLA-4 comes to the immunological synapse, this area here. Sorry, I forgot about that. So that's basically, CD28 means you're stepping on the accelerator, right? So you are going for an immune response. Now, CTLA-4 comes to this area, and at the moment that it comes to this area, it also engages with this co-receptor, and it sends an inhibitory signal. So if you think about the analogy of the car, that's equivalent to stepping on the brakes. So that's how CTLA-4 regulates immunity. CTLA-4 is so important that if you have a mouse that is deficient for that molecule, the mouse dies within 2 weeks of being born, and it dies out of autoimmunity. So it's a key regulatory molecule in the immune system. Well, so what Jim and collaborators decided to do is to make an antibody that will block CTLA-4, in a way releasing the foot from the brake, right? And he hypothesized that by doing that, the full signal will deliver into the T cell. You step on the gas, on the accelerator again, and you get anti-tumor immunity. So that's what he hypothesized. And this is data not from Jim's lab, this is data from my lab, but this has been verified in many, many papers, and he published the same thing in 1996. So if you take several mice that you challenge them, you give them a tumor, the tumor grows up to a size, and then you sacrifice them. If you give them a control antibody, a control immunoglobulin, there is no change in the growth of those tumors. However, if you give those mice 3 shots of the anti-CTLA-4 antibody, you guys can see the tumors continue growing, reach an inflection point, and then they collapse, right? And they never grow back in this mouse model. Depends on the mouse models, it will have different activities, different extent or power on the activity. And my laboratory have been working for several years trying to understand really how it works and generate better antibodies, etc. But we're not going to have time to talk about that. What I want to talk about is how do we move that into humans and what has happened, right? Because everybody will say mouse models don't translate into humans. And I think that this is one of the first demonstrations that it does. So this is a metadata analysis of more than 2,000 or about 2,000 patients treated with the anti-human CTLA-4. It's called ipilimumab. Late-stage metastatic melanoma is a disease where there's only about 10% alive after 2 years, and then pretty much everybody dies. So it's a pretty brutal disease with very little solution to it. Chemotherapy doesn't do much against late stage. And what this data slide shows for cancer patients with late-stage metastatic melanoma is that if you have that disease and you are put onto this drug, if you make it alive by year 3, there's more than a 90% chance you're going to be alive for more than 10 years. 10 years alive with late-stage metastatic melanoma, it's a game changer, right? So that didn't exist before this therapy. Obviously, there is a lot of room for improvement. This is only 20% of patients, but it's 20% of patients with long-term survival. This changed the way we all think about cancer therapeutics. So this opened the door to say, actually, immunotherapies have something. It can be improved, but they have something. And obviously, you can start asking, well, is CTLA-4 the only relevant molecule that we could target or tamper with to generate a potent anti-tumor immunity? And the answer is no. The beautiful thing about biology is that once you find a molecule, you find families of molecules with similar activities. And that's how Takusho Honjo, in the 90s, discovered this other molecule called PD-1. And then Liping Cheng, in the early 2000s, discovered the ligand for that molecule that is called PD-L1, PD-1 ligand. And in a shorter period of time, you see how the curves start getting closer together. Drugs that target those two molecules have been approved by the FDA and the EMA against metastatic melanoma, against lung cancer for the FDA. So there are more drugs on the pipeline. How does PD-L1 work? Well, so PD-L1 is called the molecular shield. If we have our bad guys here, the tumor, let's say we activate an immune response, we get a lot of lymphocytes inside, so that would be a good thing. But at the moment that the lymphocytes get inside, the tumor senses the presence of the lymphocytes, and it puts up this shield, and the T cells cannot work, and they stop. And actually, many of them die. If we look at it in another type of cartoon, what happens in the tumor, now we have the tumor cell, the T cell, the specific recognition, and PD-1 is on the cell surface of this activated T cell. So because it's on the cell surface, when it binds to PD-L1 expressed on the tumor, it sends an inhibitory signal. So it prevents the full activation of the T cell at the tumor site. And that, again, is like stepping on the brakes. So what Takusho Honjo and Liping Cheng and many others have done is the same rationale. We're going to block this immune checkpoint with antibodies either for the ligand or the receptor, release the brakes, right, and step on the accelerator, and that actually should generate a very potent anti-tumor response. So that has been tried as a monotherapy in mouse models as well as in clinical trials and gave very promising results. There's not enough time to look at all of them. But I just want to show you the question of two combined molecules. So that's where we are. We're combining now CTLA-4, anti-CTLA-4 and anti-PD-1 antibodies. And are two better than one? Well, the answer is yes. So this is one of the first clinical trials where they combine the anti-CTLA-4 antibody with the anti-PD-1 antibody in patients with late-stage metastatic melanoma. This is called a spider graph. It's a very striking graph to show because the numbers going up means the average size of all the tumors on that patient. Each line is a patient. So if it goes up, it means that the tumors are growing. If it goes down, it means those tumors are crashing on site on a patient. This is not a mouse model. And if it goes down more than 80%, it means that more than 80% of the overall tumor mass of that patient disappeared. And what's very important here is the length of this curve. It's not just an ablation of the tumor, it's a durable response, durable regression of the tumors. This is another way of showing some of the data coming from these trials. This is the historical survival in patients with late-stage metastatic melanoma. So the more you drop, the less patients you have alive. This is the time in months. It's about 10% overall survival at 2 years. This is an experiment, not an experiment, a clinical trial where they were testing different amounts of the anti-PD-1 and anti-CTLA-4 data, antibodies. This is at the lower doses. So the 2-year overall survival is 50%. 50% of the patients alive, 79, 88, and that's still ongoing. So obviously, we expect these curves to crash over time. But if they follow the trend of anti-CTLA-4, what people are expecting is actually stabilization of the curve every time a little bit higher. So there is increase in patient survival by combining these two drugs. Now, the question that I hope you all guys are all asking is, well, you're releasing the brakes on the immune system and you're stepping on the accelerator. So what happens with my tight regulation, my tight control? I thought those molecules were there to protect us from autoimmunity or auto-attack. And that's what they are there. They are to protect us from that. And actually, the side effect of using these drugs is autoimmunity. And these are some of the examples of autoimmune pathologies or adverse immune events associated to different immunotherapies, particularly the CTLA-4 and the PD-1 targeting pathologies. So we have several of them. One of the most important ones is colitis, like high-grade colitis. These are controllables by using steroids, anti-inflammatory drugs, but they are a problem. And we need to understand much better how to combine these drugs in order to maximize anti-tumor activity and reducing toxicity. But if you talk to clinicians giving these drugs, the toxicity is manageable and survival is improved. Now, beyond CTLA-4 and PD-1, it becomes even more complex. There's a large and growing, ever-growing pipeline of these immunotherapeutics. So I just show you two of them, CTLA-4 and PD-1. If this is a cell, a T cell, it receives a specific signal through the T cell receptor to, remember, stop and think, what should I do? CTLA-4 and PD-1 will make sure to inhibit T cell activation. In the same way, TIM3, BTLA, VISTA, LAG3, etc. This is a slide from 2011, so it's quite outdated. They will also, there are also receptors on the surface of the T cell, and they will send inhibitory signals. The guys on this side, they will deliver positive signals into a T cell. So you can imagine how complicated this is. This is pretty much like a control board, like a very, very complex control board, where you move your dials, right? You give positive signals, inhibitory signals, and what the immune system evolved to do is to give all of that to basically avoid immunosuppression, right? So if you turn everything down, you're not going to get a response. If you activate everything, you're going to get autoimmunity. And what your immune system evolved to do, which was on the slides of the boy on top of the cesspool, is to have a well-tuned and perfect immunity. And that happens because of the interplays of these positive and negative signals. Now, what cancer immunologists have done, they've looked at this, and they generated now a large pipeline of antibodies that can, just like CTLA-4 and PD-1, block inhibitory signals into a T cell, or deliver positive signals into the T cells. And these are molecules, the vast majority of these are already in clinical trials. There's not enough data to make conclusions like the ones we can make for CTLA-4 and PD-1. But the point of this slide is to show you how much more work we have ahead of us in terms of finding out how these molecules work, not only in preclinical setting, but also finding out what's the best way of delivering them and actually preventing autoimmunity. Now, in the context of autoimmunity and trying to prevent autoimmunity or trying to give more direction to this response that we have released the brakes on and step on the accelerator, we go into a topic called recognition. And something that I haven't discussed with you guys is a key question in the immune system. What is it that T cells recognize on tumors? Why is it that T cell, when I said it stops why? Why is it recognizing that it makes that cell so appealing? Why is that important? Well, because if you think about tumors and healthy tissue has self-antigens. The immune system evolved not to recognize self. That's because it wants to avoid autoimmunity. So what we want is really target those molecules, those antigenic determinants of tumors. But the problem is that when we give the immunotherapies that I described, we step on the accelerator for the responses against those two types of tissues. Right? There is no rationale why would you prefer one or the other. So that's what results in toxicities. Now, if we go back to that timeline, there is actually a very interesting blast from the past. In the 90s, Thomas Welfel, Thierry Boone, and Hans Schreiber, they hypothesized that those same mutations that make a tumor, a normal cell transform and become a tumor, because there are mutations and they are non-self, they could be recognized by the immune system as something foreigner. So the same mutations that can give survival advantage to a tumor could be the cancer's Achilles heel, right? Because only the tumor will express that mutation. So going back to this graph, healthy tissue will express self-antigen, but the tumor, besides expressing self-antigen, will also express mutations. And those could be specifically recognized by the immune system, in a way allowing us to, if we could make vaccines to these guys against those mutations, just change the immune response on those T cells that recognize the tumor and not healthy tissue. So this has been a holy grail for a long time. For a long time, into Tim Chan, Bob Schreiber, and Tom Schumacher resuscitated the whole field of mutation landscape. They took the idea from Thomas Wolfe and they said, well, let's test this if this is true. Let's test whether mutations are associated to better outcomes. Mutations in your cancer are associated to better outcomes in patients with cancer treated with immunotherapy. So what Tim Chan did in Memorial Sloan Kettering, he took patients with melanoma that had been treated with anti-CTLA-4, and he asked, is there a survival advantage if your tumor has more mutations? And the answer was quite striking. There is a huge survival advantage on those patients that their tumors were more visible to the immune system because they have accumulated more mutations. So mutations are bad. Remember, this is a complex system. Mutations are bad, obviously, because they can transform you. But in a way, when you are in that balance between immune cells and cancer, mutations are the substrate, or at least one of the substrates of recognition of the tumor by the immune system. And that's shown in this slide. So this has generated a lot of enthusiasm for personalized therapies, because you can imagine now that you could take a patient, get a biopsy of their tumor, sequence, look for mutations, and based on those mutations, make a vaccine, combine it with a checkpoint, etc. And there are 3 or 4 companies already doing clinical trials with this. But there is an elephant in the room that has only been recently addressed. Tumors are not homogeneous masses. They're quite heterogeneous. And they evolve in a Darwinian manner, and this has been demonstrated by Tarek Ember and others in hematological malignancies, and by Charlie Swanton at UCL in solid cancers. Basically, if they evolve as Darwinian evolution does, you have the tumor will grow and develop first as an homogeneous mass. So this is a normal cell. The normal cell will accumulate mutations, many mutations that are irrelevant, until it finds a mutation that transforms, KRAS, BRAF, etc. The moment that it transforms, that whole tumor mass, it's homogeneous. It's perfectly homogeneous. But let's say this is lung cancer. You have another cigarette, and you accumulate some other mutations, and those mutations will not be expressed by all tumors. You have another cigarette, and you accumulate some more mutations, another one, some more mutations. As you're doing that, you're increasing the heterogeneity, you're increasing the complexity of your tumor. And the question is how those little T cells, right, running around, are going to deal with that extreme level of complexity. And that's a question that has been running in the field for quite a while. And we started a collaboration with Charlie Swanton's team. They were doing the bioinformatics, trying to understand how the tumor evolved, and our team was doing the immunology, trying to understand how the immune system will deal with that mutation. And what we find out is that basically what you have is areas of the tumor that share these trunk mutations, these mutations that are in every single tumor cell. And that there are mutations that are only on the branches of cancer evolutionary tree. So they're particular for some areas. So through a lot of bioinformatics and wet lab science, we managed to show that actually the immune system cares mostly about those truncal mutations. And that if you were to generate vaccines or therapeutics, they should be targeting the trunk of cancer's evolutionary tree instead of the branches. And one of the demonstrations came out from this data set, where we took patients treated with anti-PD-1. These are lung cancer patients that were treated with anti-PD-1, and we asked whether the patients that have a high number of these truncal mutations survive better or not. And the answer was yes. So the more mutations you have, and the more homogeneous your tumor is, the simpler for the immune system to attack that tumor. So that can be represented here. So this will be an easier to treat tumor for the immune system, and this will be a harder to treat tumor. Now, what we learn from these studies is that the immune system appears to be also distracted by the branches of the tumor evolving, the evolving tumor. And actually, what you need to do again is to target those mutations at the trunk. Truncal mutations are key, and we can find plenty of T cells within the tumor of patients with lung cancer that target, that binds and recognize those truncal mutations. And what's more important, we did several other analyses on those cells, and we find out that they express very high levels of the PD-1 molecule. So although they are there in the tumor, they can recognize the tumor mutation, they are incapable of making or performing tumor destruction because they're being regulated by this molecule. Hence, it makes sense to combine therapies that target those T cells or activate those T cells with anti-PD-1 therapies. So we believe that based on this data, what we need to do is to activate T cells that target those truncal mutations so they don't get distracted with the branch. If you target just the branch, right, you're going to be basically pruning the tree of cancer's evolutionary tree. What you want to do, if you go back to the analogy of the tree, is you take it from the bottom. Right? You generate a therapy that can target several truncal mutations. So the whole cancer is destroyed. Think about it again as a tree. If you make a vaccine to a mutation that is only expressed by parts of your tumor, you're going to take that down. That's great. But the rest is going to keep on growing. Right? So you're only going to be pruning that tree. And just to close, to give you a general idea of how would you mobilize these trunk reactive T cells, well, you can go to the vaccine field, the same field that I told you that there are already clinical trials, where people take a patient with different cancers, let's say in this case lung cancer, you take a tumor, and you can sequence that tumor using the current technology. But then you can use different fancy bioinformatic tricks to identify the trunk of that cancer, right? The trunk of cancer's evolutionary tree, and make peptide vaccines, DNA vaccines, or RNA vaccines, whatever is your favorite platform, but vaccines that will make the T cells activated only against those truncal mutations. Now, if any of the things that I said on the first part of my class are clear, you should believe that this will never work, as it is on the board. And it will never work because this is only the specificity signal. This will only tell the T cells, go and attack. And the T cells will go, they will find the tumor, and they will stop, and they will not do anything. They will not do anything because we need to tamper regulation. So what really want you guys to take home today is that T cells are important. They make it into the tumor, but the fact that they're there doesn't mean that they will be able to eliminate that tumor. The tumor will take over immune regulation and will shut down T cell function. And we can use immunotherapeutic approaches such as antibodies, but there are many other ways, to tamper with that regulation and fully mobilize or fully activate T cells to destroy the tumor. In a way, by making vaccines to the trunk, now we're steering that car, right? We're not just releasing the brakes and stepping on the accelerator. We're giving direction to anti-tumor response. And what we hope with this type of approaches that haven't been done yet is to obviously try to maximize responses and minimize toxicities. So key concepts to take home, and this is just pretty much a repetition of what I've been saying, is that there is a crosstalk within tumors, a crosstalk between cancer and the immune system. And that crosstalk or equilibrium is heavily regulated. But it can be interfered, it can be tampered with immunomodulatory agents, such as antibodies. And finally, we can give direction to the response that we're deregulating by using at least different ways, but one of them will be vaccination approaches, and most importantly, against mutations that are shared across every single tumor cell. And based on everything, the most important message to take home really is that we can mobilize the immune system to fight cancer. I just gave you a couple examples. There are many more than that. They can impact patient survival, and there is still boatloads to learn. So there is topic for everything. Everything is coming back. Radiotherapy, chemotherapy, they're all being reassessed in the context of immune regulation, in the context of vaccines. So we're going to take that whole timeline, and we're going to revisit pretty much every single thing that has been discovered in the context of immune regulation. And that's going to be probably very interesting to see evolve. I didn't show all the data of the work done in my lab, but this is my lab. I want to acknowledge the people that fund my work, which is Cancer Research UK mainly, Cancer Research Institute, Sam Keane, the Royal Marsden, and several others. And the last bit that I show, the last bit of data, the one about cancer evolution, it's really an example of trying to work at the interface between cancer biology and immunobiology. And we're seeing this more and more today. We're seeing that those papers that were one PI and one postdoc, we don't see many of those. Now we see papers with 30, 40 people, because these are huge endeavors, huge collaborative endeavors. And I have to hopefully motivate you guys to collaborate, because that's really the only way to innovate. If we keep on doing what we're doing without changing, without innovating, without collaborating, we're not going to get anywhere. And with that, I'll take any questions.
Okay, so we open for questions. Okay.
What are the factors that push the system away from the first phase of equilibrium between the,
What prevents that, the first phase?
No, no, sorry. When basically you said the three phases. So when the immune system is able to control potential cancer growth, and then there is a battle, and then the tumor wins.
Yeah.
But so, what are the elements that tips the balance in favor of the tumor?
Yeah. So let's say it's a viral-driven tumor. It's a viral-driven cancer. Because there is a very strong signal 1, right? The immune system doesn't need that much danger signal, that many danger signals. It's all a balance, and it's able to eliminate the tumor. So that's one of the ways that you switch it to elimination. Now, how do you get to the equilibrium and then to the escape? The tumor, because the tumor replicates a phenomenon called wound healing. So as the tumor develops and accumulates more and more mutations, the same pathways that are altered by those mutations make the tumor look like a piece of your body that is trying to heal. And when it's trying to heal, it's trying to shut down inflammation. So although you could still be recognized by the cell, by the T cell, by the T cell receptor interaction, the signal 2, these danger or inflammatory signals, are downregulated. And there are several ways of doing that. So for example, the tumor could start secreting anti-inflammatory cytokines like TGF-beta and IL-10. The tumor, via the same mechanism of angiogenesis, can shield itself from T cell infiltration. Right? So those are two of many different mechanisms. The tumor will also recruit a subset of cells that I didn't talk about that are called regulatory cells, that they can infiltrate, sit next to a tumor, and whenever you have a T cell trying to destroy it, the regulatory says, no, can't touch this.
Are there lifestyle lessons, let's say?
Smoking induces cancer. I think we all agree. Diet, there are concerns about diet. I'm not a population geneticist, so I really can't speak a lot about it, but I think diet will affect your immune system. Type of foods that you ingest can affect cancers that are of the gastric system, right? So if you eat a lot of stuff that is carbonized, like, I shouldn't say this because I love barbecue, but lots of barbecue and all that, like epoxides, DNA modification. So yes, lifestyle can definitely affect you, but the best example is smoking.
Yeah, do you have any questions? I can make a question then. Do you know how much money is spent in research, the more traditional research for cancer and in immunotherapy, just to have an idea of how much this has grown in the past?
That's a very good question.
Yeah.
The number is not enough. I don't know. I just know that there is a big effort in the UK to fund a lot of cancer immunology. So depending on the funding agency, for example, Sarah UK, I know that they have, one of their initiatives is to develop more immunology and actually make natural immunologists that are not doing cancer to start working on cancer. So it is significantly improving.
Oh, there is one question. Do we have time for one more question? Last question.
I'm just wondering to what extent you're getting support from pharmaceutical companies, because of course pharmaceutical companies have got vested interest in the status quo.
That's a great question. Unfortunately, I don't have much time to answer it. I have 2 grants from pharmaceutical companies. It used to be that pharmaceutical companies will just take from the scientists and run away, or that they would not consider the scientists at all. But academia has a large power. So in academia, we can systematically analyze mechanisms of action. And by understanding mechanisms, you understand why drugs work and don't work. So pharma now, it's aligned much better with academia. So it looks very interesting. Smart pharma is aligned with academia. Thank you.
Thank you.
Thank you very much.